For most of human experience, we lived in societies where past, present, and future was about the same. But a couple of hundred years ago, we began to live in worlds where there was such rapid change that people became uncertain. We are moving into an era of technological change that is well beyond anything we ever had before.
Would that be okay?
Our hope was that if we could understand the gene, that might provide the foundation for effective forms of therapy.
Just like you can change a single letter in a Word document, now we can change single letters in the DNA.
I believe that the consumer is capable enough to get this information on their own. Everyone could be a geneticist, everyone can understand it.
And if you're wrong, that can have huge consequences.
People said all sorts of nasty things about it. They thought they were creating designer babies, they would create monsters.
The future of medicine.
The future of healing.
The future of diagnosis.
The future of babies.
The future of drugs.
You should not assume it's going to be the way it has been. The tsunamis are coming, the waves are coming.
They're rushing in faster than we can make a decision about what's good for us and what's bad for us.
We can either ignore them and be wiped away, or we can become a surfer, study them, and surf them.
The worst thing you can do right now is nothing. Don't be the guy who refuses to look at the future.
I have this very aggressive type of cancer. It's hard to treat.
Precision medicine, it's being called the future of healthcare.
It means the right dose of the right medicine for the right patient.
A super targeted approach.
It is really like a heat-seeking missile.
Expectations are sky high for precision medicine, a new strategy that tailors treatment to individual patients.
You can match a blood transfusion to a blood type. What if matching a cancer cure to our genetic code was just as standard?
But we've heard these kinds of hopes before, during the race to sequence the human genome.
The race to crack the human genetic code, which could lead to life-saving, revolutionary changes.
Medicine will be transformed.
There's at least the potential to reduce the number of cancer deaths to zero during our lifetimes.
About 30 years ago, Marston Linehan began searching for a new way to help his patients with kidney cancer.
Now, who else in your family had kidney cancer? There were no effective forms of therapy, no drugs that worked. I lost every one of these patients who developed advanced disease. So we wanted to identify a gene for kidney cancer, and our hope was that if we could understand the gene, that might provide the foundation for effective forms of therapy.
It was the start of the genome age, and scientists around the country were hunting for disease genes.
We had no idea what a mountain this was to climb. We set up a whole program. We analyzed DNA from over 4,000 patients. It took us nearly 10 years to find that gene.
Then a group of government scientists had a big idea. Instead of searching for genes one by one, what if they could create a map that would locate and sequence every gene in our DNA?
People are mostly hoping to see this have some payoff as far as human health.
Led by Francis Collins, the Human Genome Project was the most ambitious biological research program ever attempted. It started in 1990 with a 15-year timeline and $3 billion budget.
So the motivation really was to enhance the ability to understand hereditary factors in all diseases as quickly as possible.
And the sorts of traits that affect the vast majority of the population, heart disease and diabetes, we know precious little about, and those are really the frontiers of genetic research.
At the beginning, we didn't have anything automated.
This is exon 5. This is a family.
Everything was done manually. You had to do all these chemical reactions one by one by one. There was radiation involved. You had to wear protection. It was horrible. 1, 2, 3, 4, 5.
The genome is made up of about 6 billion chemical letters. Collins knew without dramatically improved technology, it would be impossible to read them all.
We were, by 1998, finally where we could see this goal might be achievable. It was at that point that a separate effort was announced in the private sector.
This is the largest DNA decoding lab in the world by far.
Former National Institutes of Health researcher Craig Venter spent years trying to speed up gene discovery.
I was the first to make the transition from the old manual way. My lab had the very first automated DNA sequencers.
His announcement that his company, Celera Genomics, would sequence the genome before the government shook the scientific world.
It's going to be years, if not a decade faster.
He was using a new approach called shotgun sequencing, where the genome is broken up, sequenced, and quickly reassembled by a computer.
It's like a jigsaw puzzle with 25,000 pieces. But we knew how to put the pieces together.
Suddenly, it was a competition. Venter suggested that Collins' team sequence the mouse genome, leaving humans to him. Collins refused and said Venter's untested approach was likely to produce a genome full of gaps or mistakes.
It's not just about science, but also about the race for vast sums of money.
Collins also attacked Celera's plan to sell genome information and to patent genes, which could make it harder for medical researchers to work on them.
Somebody else who has a great idea about what that gene might do and a way in which to turn that into a product for public benefit may be discouraged from doing so.
The tension soon made headlines.
Are you going to win? We're not going to lose.
Everybody likes a good competition. It's not often that a small group takes on the whole federal government.
It actually made this effort to sequence the human genome something that people had heard about.
The greatest human science project ever.
Once it's on the front page of the New York Times that there's a competition going on, and then everybody is like, what's going on there?
Every day, it's robot versus robot, public computer versus private computer.
Both the public program and Celera were, at a frenetic pace of sequencing. It became clear that the acrimony was only going to grow. I was starting to become concerned that this was turning into a mud wrestling match. And it was damaging both of them, and of course damaging to our program as well.
One of the Genome Project's original architects, Department of Energy scientist Aristides Petrinos, arranged months of secret meetings between Venter and Collins, hoping to make peace. And on June 26, 2000,
I am happy that today, the only race we are talking about is the human race.
They presented the genome to the world together.
In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer by attacking their genetic roots.
For researchers like Marston Linehan, hoping to use genes to find cures, it was like striking gold.
The Genome Project gave us the map. All of a sudden, we knew the sequence of every gene. We didn't have to spend years and years finding them. That was a huge advantage for us.
But it also made things more complicated. After finding one kidney cancer gene, he kept finding more and more.
We know of 16 genes that cause kidney cancer. We thought kidney cancer was a single disease. It's not a single disease. It's a number of different types of cancer that just happen to occur in the kidney.
And who is that?
You? Alex Din was only 34 when he got kidney cancer.
Look, this is when I first came to America. I was born in Vietnam. My parents always wanted to give me a better life. The week that I was diagnosed, we just found out that we were expecting our third kid. So it was, this week of roller coaster emotion. I had my left kidney removed, and then it started to spread to the other side of my abdomen. So then we said surgery is not an option anymore.
Alex and his wife are doctors. Wondering why he got cancer so young, they went for genetic testing.
They tested the entire genome and found that I had this one specific mutation. Rare gene, kidney cancer is rare, I get them both.
Through a friend, Alex heard about Dr. Linehan, whose lab was using genes to develop treatments for kidney cancer.
Once you discover a cancer gene, we take tumors from patients and we study how the cancer gene works. And then we work on the development of drugs.
In this case, drugs for the type of tumor Alex has.
And when you go here after treatment, it's all dead.
And if they look good, then we go to clinical trials in patients.
How are you doing today?
Good, thanks.
7 years ago, Alex joined one of those trials for his specific cancer, one of a small but growing number of cancers that can be treated with this type of targeted drug.
I'm still able to take care of my family. I'm still able to work. I mean, without it, I know I wouldn't be here still.
It's all you might call personalized medicine. It's targeting that specific gene mutation instead of just trying this drug and that drug and this drug.
But critics like epidemiologist Nigel Paneth point out that few patients are likely to be as fortunate as Alex.
We haven't yet found very many examples of drug interventions that depend upon genes. There are one or two here. So for the last 60 or 70 years, we've added about 10 or 11 years of life expectancy in the US. None of that is due to genetic discovery.
That's because, after years of research, it turns out that the relationship between genes and most serious health conditions is more complicated than expected.
Heart disease, schizophrenia, some of the common cancers. There was a hope that while those would not be the result of a single gene, they'd be influenced by a short list of genes. Maybe for diabetes, there would be, 4 or 5 genes.
But we're already over 400.
Each one of which has a very modest effect, and we didn't know that until we looked.
Good.
Francis Collins, whose own lab works on diabetes, still wants to find out what role genes play in most common diseases.
Francis Collins of NIH, here.
As head of the National Institutes of Health, he's helped start a massive new federal effort called the Precision Medicine Initiative.
So the time's right to unleash a new wave of advances in this area, in precision medicine.
It has employed dramatic advances in sequencing.
The year Dr. Collins helped sequence the first human genome, it cost about $100 million, and today it costs less than $2,000.
And supercomputers to sift through the genomes and health records of about 1 million people, searching for the links between genes, the environment, and our health.
Their genes and their environment, and they somehow come together in a way that causes somebody to become ill or not. So if we really wanted to get a complete understanding of what causes diabetes or the common cancers or Alzheimer's disease or mental illness, you got to look at both of those things.
But Collins and the government still have competition.
We're processing, on average, a complete human genome every 15 minutes. And we set a target goal of trying to get 1 million genomes by 2020.
In 2013, Craig Venter founded Human Longevity, one of several private companies also analyzing genomes and health data, hoping to pick up where the original genome sequence left off.
It's just not a direct linear line of the genome was sequenced, we found this gene, and now X. And what we're discovering now are the things that occur maybe once in the human population out of 10,000 or 20,000. It's the rare combinations of things that determine our human traits.
But it's hard to know how much impact those discoveries might have.
Huge amounts of data don't guarantee that you will find the things that matter. In fact, they increase the chances that you will find tiny things that don't matter very much. It won't be a solution to diabetes, hypertension, heart disease, lung cancer, breast cancer, because those are not largely determined by the genetic inheritance we get.
For those who've been part of the genomic revolution,
without these medicines, I'm sure I wouldn't be alive today.
There's no question but to push forward.
Well, it's like we're coming out of the dark ages. Medicine's been based on rough symptomology and a little bit of hocus pocus without knowing the true molecular nature of what's going on.
There's the famous first law of technology, which is when something truly significant is discovered, its consequences are always overestimated in the short run and underestimated in the long run. I think that's absolutely true for the human genome.
This is a milestone that could one day erase hereditary conditions.
Just like you can change a single letter in a Word document, now we can change single letters in the DNA.
It's called CRISPR.
Clustered, regularly interspaced, short palindromic repeats.
In recent years, the new gene editing platform, CRISPR, has generated a lot of excitement.
CRISPR could help rid us of disease.
But this isn't the first time scientists have tried to fix the genes that make us sick. It started over 30 years ago.
Gene therapy.
The medicine's new frontier.
It was the first time they were going to be altering a human's DNA.
The expectations were crazy.
I like reading as well as playing chess, and I like to go to different places.
I would want to make people more aware about sickle cell.
How are you, Pierce?
Pierston Charles has sickle cell anemia. It's a painful genetic disease that distorts his red blood cells so they can't deliver oxygen to his organs.
His first blood transfusion was 7 months old. He had a stroke. He lost his spleen. And shortly after, the gallbladder. He's had a total of 48 hospitalizations.
He's one of about 100,000 people in the United States with sickle cell. Most are African American and on average live to their mid-40s.
There's no definite treatment for sickle cell.
Sickle cell was one of the first hereditary diseases to have its gene mutation, or sequence change, identified.
Ever since I met my first patient with sickle cell disease, that's been really my challenge to say, let's find a cure that is based on the cause. We've known the sequence change since the early 1970s, but I'll point out that's over 40 years ago, and we don't have any therapy based on knowing that sequence change.
So in his lab at Stanford University, Matthew Porteous is trying to repair the gene that causes sickle cell.
So what we have to do is edit some blood stem cells.
He's using a new gene editing technology called CRISPR-Cas9.
Ready to electroporate.
It works like a genetic scissors, cutting out and replacing a gene's misspelled bits of DNA.
We can change single letters in the DNA and convert the disease-causing variant to a non-disease-causing variant. The cells that have been edited are these bright green cells.
Pierston needs monthly treatments to replace his sickle-shaped red blood cells with normal cells. He donates the old blood for Porteous's research, which he hopes could someday help him.
They're attempting to modify the genes so they can cure sickle cell. I think that it's pretty amazing.
Porteous is preparing to start a clinical trial.
We think these cells represent cures for patients. They do everything that they're supposed to do. So that gives us confidence that it's worth trying in a human patient.
We have our fingers crossed. We're excited.
But along with CRISPR's promise comes fears about its accuracy.
A DNA break could be dangerous. It could cause other mutations.
And its potential use editing genes not just in patients but in embryos, which would impact future generations.
We're talking about something that would affect human evolution.
Critics worry CRISPR could be used to create designer babies.
You may want to make a baby taller, faster, smarter.
The power of this new CRISPR technology has resurfaced questions that people have been thinking about for 40 years. If we had a tool that would allow you to change the DNA that would then be passed along to next generations, is that something we should or shouldn't do?
Many of the questions and the controversy remind Ashanti De Silva of her own experience with a different kind of genetic engineering called gene therapy nearly 30 years ago.
Ashanti, come baby.
When I was born, the umbilical cord was infected and I would turn blue a lot.
She was born with ADA deficiency, a rare genetic disorder that crippled her immune system. Though there was no cure, her doctor had an idea, though it could take a lifetime.
He said, there's this potential trial going on at the National Institutes of Health. Let me send her blood.
And he told me, Mrs. De Silva, this is for possible gene therapy when your daughter is 60 years old. And I thought, oh, how wonderful.
Unlike CRISPR, which repairs genes, gene therapy would send in an extra healthy gene to do the work of the defective one. But before Ashanti could be treated, there were scientific and ethical hurdles to overcome.
They had told my parents there'd be risk of leukemia, death. It was the first time they were going to be altering a human's DNA. We all went to the NIH, and they actually didn't have FDA approval yet. It was really up until that day. They were waiting for the phone call. I was in the room.
This is on the 14th of September. A great day for the world, a great day for medicine. Gene therapy has been approved. You can read that.
Our daughter is the first patient in the world to receive gene therapy.
They had taken my cells and injected the vector with the corrected gene into my cells. They transferred those cells back to me to see if they would give me a new immune system.
How about that? Okay.
My parents did receive death threats because there was this idea that altering someone's DNA is playing God.
There was a historic medical first today. Genetic engineering as a potential way to cure an illness in a human being.
The gene therapy revolution seemed to come almost instantly, fueled in part by discoveries from the Human Genome Project, the national effort to find and sequence all human genes.
Payoff has already begun by the discovery of a long list of genes.
As researchers found more and more of the genes that cause hereditary diseases,
it is only a step toward eventual treatment, but it is a significant one.
Gene in a cancer patient.
News stories regularly touted gene therapy as the potential cure.
Gene therapy to improve a potentially deadly condition,
such as muscular dystrophy, sickle cell anemia,
and perhaps even AIDS.
We presumed as a society that if we had the gene, we'd be able to then quickly move to a treatment. The expectations were crazy.
James Wilson was one of gene therapy's pioneers.
In these human cells, he was able to replace the defective cystic fibrosis gene with a normal gene.
In 1999 at the University of Pennsylvania, he began testing a gene therapy for a rare liver disease that mostly affects boys.
Upon their first meal, they develop a coma, and there's 50% mortality as a result of that.
To deliver the healthy gene into the patient's body, Wilson used a specially prepared virus called a vector.
A young man by the name of Jesse Gelsinger received the vector. Things went terribly wrong. He mounted this enormous immune response that led to his death. I had spent almost 10 years trying to make it safe. That's probably irrelevant. What is relevant is the young man died.
After hundreds of gene therapy experiments, Gelsinger's death came as an enormous shock.
Every researcher in America was reminded this is messing with the code of life.
Why aren't they reporting these?
Congress held hearings, and federal agencies investigated.
The FDA says Penn researchers failed to report previous toxic reactions in humans and the death of test animals in similar experiments.
I wasn't given all the information, and some of the information I was given was not true.
Penn's program was shut down, and Wilson banned from clinical trials for 5 years. Then, investigators began uncovering problems at other institutions around the country.
Several others died. Hundreds more had side effects, but researchers didn't tell federal health officials.
Gene therapy also faced scientific setbacks.
A cystic fibrosis treatment, a muscular dystrophy treatment, a brain cancer treatment all failed. To this day, gene therapy has yet to cure anyone.
Including Ashanti Da Silva, the benefits from her gene therapy diminished with time, so she still needed regular enzyme injections to support her immune system.
Gene therapy, at least initially, did give me that boost that I needed. But it wasn't what they could call a cure.
The tragic death of Jesse Gelsinger was one of several events that led to a precipitous decline in the support for this field. Support just evaporated after that.
It seemed that gene therapy's prospects were over.
Here's an area of the liver.
But a small group of researchers continued on.
Our first responsibility to everyone, including Jesse Gelsinger, was to try to figure out what happened and why.
Can you put on our low magnification?
We learned that the problem was the immune response, not to the gene, but to the delivery vehicle itself.
Wilson returned to the lab, hoping to make safer vectors.
And the vectors that we generated seemed not only really safe, but were much more efficient. I knew that they would be a game changer, and we just distributed these to my colleagues. And I hoped that there would get traction. It didn't take long to discover them. It took a lot longer for there to get traction.
An experimental gene therapy is...
20 years later, gene therapy has been experiencing a revival and is showing promise for conditions ranging from hemophilia to Parkinson's disease.
One more.
Peter McConnell has a genetic eye disease called choroideremia.
It leads to tunnel vision, in some cases blindness.
He's the father of 2 young children, and hoping to see them grow up, joined a clinical trial at the University of Pennsylvania.
When I initially was approved, I didn't think twice about it. I was just going to do it. They take a genetically mutated virus and they inject it in the back of your retina. fourth letter, maybe a V.
The goal isn't to miraculously restore eyesight. What they would consider successful was stopping the eye from progressing and getting worse. I'd be more than happy if that was the case.
For the first time ever, the FDA has just approved a gene therapy treatment.
In 2018, the first gene therapy was approved in the United States for a form of childhood leukemia. But because gene therapy doesn't actually fix broken genes, no one knows how long its benefits will last.
What we're asking to do here is to cure diseases, and whether we can ultimately achieve a permanent genetic graft that reverses their disease forever, we'll see.
That hope for a permanent cure is why so many are excited about CRISPR.
Wow, human genetic engineering. Yesterday was science fiction, and today it's reality.
Get this, it could eliminate chemotherapy.
It's fast, cheap, and can cut and paste genetic code with great precision.
All the results say that it is working. The ultimate test is going to be those first few patients. They're the ones taking all the risks.
Patients who take risks like Ashanti Da Silva did with gene therapy. She's grateful for the treatment she got, but regrets gene therapy's long hiatus.
When you're doing something like altering someone's, DNA, there are risks, but it gave me life. I was able to complete my master's, get married, have a nice home life, so I've been very lucky.
As for James Wilson, he is now working with CRISPR, but remains mindful of the lessons from gene therapy's early days.
That period of exuberance early on in the development of a new, novel, cutting-edge technology, it's very easy to get caught up into that. The reality is this is still science, and these are still experiments, and we are still learning.
The genetic testing industry is booming.
The more you know about your DNA,
the more you know about yourself.
There is a simple genetic test that could show you whether you're likely to get a serious disease.
DNA tests are now being sold for everything from lactose intolerance
farewell dairy
to cancer.
It's like a cancer stethoscope.
But how well do they predict the future?
I could get stomach cancer. There's an 83% chance of it.
I definitely thought you have the gene or you don't have the gene. I did not think there was any gray area.
Excited for class today?
Yep,
I can feel the energy.
There were 5 kids in my family. My brother Steve is the one who had the mutation and passed away.
Massachusetts teacher Beth Lambert lost her brother in 2006. He had stomach cancer caused by a rare mutation in a gene called CDH1.
The next step was for all of us to be tested. There are 4 remaining children in my family. 3 of us tested positive for the mutation, and we knew our choice was to have our stomachs removed. If I didn't do this, I could get stomach cancer. There was an 83% chance of it.
How's your day been?
I was 38 years old, married. I have 2 kids. It was not a difficult decision because we watched our brother suffer.
For Beth and her family, the promise of genetic testing is clear.
1% of stomach cancers are of this type. At that point, I think only about 100 families in the world even had it. There are so many people who have a genetic condition and there's no solution. I feel so grateful that we have a choice.
Life-saving genetic tests were among the things scientists hoped for when they sequenced the human genome in the 1990s.
It'll give you power over your own life instead of just randomly waiting for symptoms to appear.
By the time the Human Genome Project was unveiled in June of 2000, it seemed as if testing would soon be available for almost any ailment.
We are going to know why I'm at risk for one thing and you're at risk for another.
Silicon Valley pioneer Ann Wojcicki is a big believer in the power of genetic testing to keep us well.
What I want to know is what's the best way for me to live? Like, what am I at risk for? How should I modify what I'm doing so I can be as healthy as possible?
In 2006, with backing from Google, she started 23andMe, selling direct-to-consumer DNA test kits.
I believe that the consumer is capable enough to get this information on their own. Everyone could be a geneticist. Everyone can understand it. This is what you get in the mail. It's super easy. You get the little tube, you spit in here. We tell people what the variations are in their genes. The first product was 12 reports. How much Neanderthal do you have? Which parts of the world do you come from? Prostate cancer risks, Alzheimer's.
23andMe saved my life.
While 23andMe sold directly to consumers,
the Neogen company,
the use of DNA tests by doctors was also growing.
Breast cancer runs in my family.
BRCA 1 and 2 screening for breast and ovarian cancer is one of genetic testing's best-known success stories.
The actress Angelina Jolie went public about her choice in an...
To avoid cancer, women who test positive can choose extra monitoring or to have their breasts or ovaries removed.
As for Angelina Jolie, her risk of breast cancer fell from 87% to less than 5%.
Kelly Donahue was diagnosed with breast cancer when she was 37 years old.
I had the most wonderful life you could possibly have. And this kind of put my world to a stop. Did you give the boxes? They sent me for our genetic testing. If you test positive for either BRCA1 or BRCA2, the recommendation is to have a double mastectomy, as well as a removal of your ovaries.
The colors of Kwanzaa.
I do remember getting the phone call and saying, oh, what's the news? Is it positive or it's negative? Well, it's not positive, Kelly, and it's not negative. You have a variant of unknown significance. And so now, what does that mean?
We've been collecting data on the BRCA1 and 2 genes, and there are hundreds of variants in those genes that we don't understand today, that no one understands.
Geneticist Heidi Rehm runs ClinGen, a data project that analyzes test results and symptoms from thousands of people, hoping to figure out what gene variants or mutations mean.
When we sequence an individual, they have about 5 million variants. That means 5 million spots where they're different than someone else. And the big challenge is trying to figure out which ones are actually going to cause disease and which ones have no clinical impact. If you're wrong, that can have huge consequences. There are patients that may have had their breasts removed unnecessarily. There are patients that could have had a device implanted in their heart. That patient may never have been at risk.
Kelly's gene test was inconclusive, but because she did have breast cancer, her doctor agreed to a double mastectomy.
But now, I had to worry about my ovaries. I didn't want to hear 5 years later, oh, you know that variant that you have? Well, now it's positive. And then, already have cancer in my ovaries. In the back of my mind, I had this worry. So I just kept going to appointment after appointment until finally they said, okay, we will take your ovaries out.
Hi. Good to see you.
Harvard geneticist Robert Greene studies how genetic tests are perceived and used. He says Kelly's decision reflects long-held beliefs.
Thinking about having multiple children, we went and had ourselves genetic tested.
The early low-hanging fruit were the diseases where if you had a mutation, you definitely got the disease.
Tay-Sachs testing will be held in your community soon.
And this gave us a narrative in genetics that we could do this for many, many types of diseases. But it's just much more complicated than we thought.
Okay.
In recent years, Greene has tested healthy people and found that sometimes they can carry a well-known mutation that we expect to be dangerous without getting sick.
You have this mutation for the disease chondrodysplasia punctata. And yet you have had no complaints that would be related to this condition.
I'm almost 60. There's no symptoms, so no, I don't worry about it.
He had a mutation which is associated with a bone disease. Now, he doesn't have a bone disease.
For doctors and patients, it's hard to know what to do with these kinds of results.
What happens if you have a mutation for a cardiomyopathy, a thickened heart wall, but you've never had a symptom in your life? Should I be concerned? It's a completely unknown question.
Greene cautions that testing's ability to predict future health may be most limited when it comes to the conditions we most worry about.
For the vast majority of heart disease, diabetes, and almost all of the common cancers, there simply isn't one gene or one gene change that's going to predict whether or not you get that.
Ironically, at the same time scientists were discovering limits to their predictions, the business of DNA testing was booming.
32 companies advertise on the internet offering to test your genome for a host of diseases.
Tests that promise to predict your future.
your risk for cancer, for other diseases.
By 2013, 23andMe had tested over 500,000 people for an increasingly wide range of ailments.
Users can find out about more than 250 health conditions and traits. The results cover everything from lactose intolerance to Alzheimer's disease.
But the Food and Drug Administration determined that 23andMe's results, along with results from other direct-to-consumer tests, were not clinically validated.
In terms of what the results mean, for many of the claims being made by these companies, the science is not yet sufficiently evolved.
The FDA then sent a scathing warning letter ordering 23andMe to halt the sales and stop.
23andMe had to stop selling health tests, and several of their competitors closed. But to save her company, Wojcicki worked with the FDA. Out of over 250 reports, the agency has approved around 10 health risk reports and a few dozen for carriers of disease genes.
We have had to change everything. We also have a limited subset of what we used to have for reports, but we are working with the FDA to try to get more reports back. They want to make sure that the data is accurate and then make sure that people understand it.
The FDA is developing new regulations for consumer medical tests. But in the meantime,
your personal DNA nutrition test.
Testing your genome is not just about diseases.
Genome is revolutionizing the pursuit of wine through genetic testing that pairs personal taste to perfect bottles.
And it's big business. Companies make money selling tests.
Unprecedented insights about your fitness potential.
And also selling users' DNA data to drug companies for research.
Part of a market that's expected to generate $600 billion globally by 2020.
23andMe, whose millions of users can choose to complete elaborate health surveys, has several lucrative partnerships.
Could be Parkinson's with one company, it's lupus with Pfizer. The combination of data from the surveys and the data from the genome will enable us to do massive, huge studies. I do think that there's a race about data and genetics and really understanding what you do with that, and everyone's running fast.
A or a G.
Whether it's for medicine or commerce, nearly 20 years after the first genome was sequenced, scientists are still trying to understand what our genes mean and how to use them to improve our health.
Hello again.
Now we're sequencing millions of genomes and learning something new with each successive one. Over time, we will understand what this information means. Our hope is that we can identify true risk for disease at a level where an intervention makes sense.
The way it did for Beth Lambert.
We were thrilled to have that information and to be able to do something about it. My brother didn't have a choice at all, and it's the greatest gift he could have given us. The surgery literally saved my life.
A revolutionary technology that can edit genetic mistakes.
The public debate over the gene editing technology CRISPR is often sidetracked by what it could mean for the future of babies.
Designer babies.
Designer babies.
Designing babies is not allowed in America now, but it's coming.
It's not the first time a scientific advance involving embryos has captured public attention.
A British medical team said today it hopes to create the world's first test tube baby by the end of this year.
In the 1970s, the idea of in vitro fertilization was still a dream, but fears of where it might lead were already taking hold.
This is one step toward further modes of manufacturing our children.
People were just generally scared. They didn't know what was going to happen. I think it was tied up with the old novel Brave New World, in which the babies there were gestated in what he called bottles.
Mark Bernard G. Inspected and approved.
To create a baby in the laboratory in a petri dish was considered not just abnormal, it was considered immoral.
Several other doctors say they are against the idea. They claim that it opens the way for mass production of babies and, as they put it, a nightmare of biological engineering.
Concerns range from, there's a slippery slope here. Once we start making life outside the womb, once we start making life in dishes, won't we wind up saying that's the best way to do it for everybody? that we're going to wind up eliminating natural reproduction.
People said all sorts of nasty things about it. They thought they were creating designer babies, they would create monsters.
There was fear that someday the techniques could be used to develop something other than a normal human being.
One MP warned of the dangers of scientific breeding becoming a reality, of a revival of Adolf Hitler's concept of a master race.
The two scientists at the forefront of the research, Patrick Steptoe and Robert Edwards, conducted their work in a secluded laboratory far away from the media spotlight.
They were doing things like disguising themselves and making sure that their cars were parked at a different location when they went to visit or do any of the work. It was really cloak and dagger.
After more than a decade of research, their controversial experiment became one of the biggest medical stories of the century.
The world's first test tube baby was born here in Britain last night.
A pink, healthy baby girl who began life in a test tube.
At birth, it came out crying its head off and in very good state, breathing very well.
Louise came out, she wasn't a Franken baby, she was healthy, she looked normal. The fact that the first human IVF that went to term resulted in a healthy baby dramatically changed perspectives on IVF.
We forget now because IVF is commonplace, but really, Louise Brown heralded hope for millions of people throughout the world.
That hope and the media's fascination generated hundreds of headlines around the globe.
When I look back on the cuttings, newspaper cuttings, and films, we couldn't come back home to Bristol for 11 to 12 days. And when we did, there were 100 journalists plus outside our little house from all over the world. It was just madness.
The birth of Louise Brown was a Nobel Prize-winning event, not just because of the technology, but because of the beauty of what it did for Louise Brown's family and for thousands and thousands, now millions of couples around the world who have been able to have children.
Dr. Mark Hughes is part of the team of scientists that took IVF to the next level. In the early 1990s, they pioneered a technique that allows doctors to screen embryos for potentially lethal diseases.
The idea is to make a diagnosis before a pregnancy ever begins, so that couples who are at high genetic risk can avoid that disease before they ever get pregnant.
It's called pre-implantation genetic diagnosis, or PGD, a procedure in which couples go through IVF even if they don't have fertility problems. Doctors then test the DNA of the embryos and only implant healthy ones.
We can say, embryo 2, 5, and 7 don't have this genetic condition, and they'll be safe to transfer.
Not long after Eden was born, we knew there was something that wasn't exactly right.
When Randy and Caroline Gold's second child, Eden, was 18 months old, she was diagnosed with mucolipidosis type 4, or ML4, an incurable genetic disease with a heartbreaking prognosis.
Kids with mucolipidosis type 4 will likely never walk, they'll never talk, they'll go blind by the time they're 12 years old, and they will have a very limited lifespan.
High five on that, girlfriend. Ah, love you.
The Golds dreamed of having a third child, but they knew that dream carried big risks.
Because Caroline and I carry the same mutation for ML4, we have a 25% risk with every pregnancy that we could have a child with that disease.
And we have a recombination.
The Golds turned to Mark Hughes, and using PGD, he was able to identify an embryo without the ML4 mutation. Today, Eden has a healthy little sister named Shai.
It was an absolute miracle.
PGD has helped thousands of families like the Golds, but it also has raised familiar concerns.
Is it leading to the creation of designer babies?
As the science advances, ethical questions about when and where to draw the line when it comes to picking and choosing only the healthiest embryos. Critics say it can become a slippery slope.
From the very first cases of embryo testing for genetic disease, the slippery slope of designer babies was in everybody's mind. Oh, we'll be testing for anything.
the use of embryo screening procedures like PGD has expanded. They can now test for hundreds of diseases and chromosomal abnormalities. Yet much of the media attention has focused on the doctors who push those boundaries.
This is the room where the magic begins.
It's called gender selection.
For over a decade, Dr. Jeffrey Steinberg has been a flashpoint in the debate, constantly in the news for marketing the use of PGD, not just for medical necessity, but to let couples choose the sex of their child.
Dr. Jeffrey Steinberg, director of fertility institutes, says up to 90% of his patients come to him specifically because they want to decide whether they have a boy or a girl.
The technology was out there, it was being applied only to preventing diseases. Well, I decided to open the door and expand it and say, listen, this is something that people are interested in, causes no harm, makes people happy, let's expand it.
Sex selection for non-medical reasons is illegal in many countries, but not in the United States, where some aspects of the fertility industry are loosely regulated. Many of the procedures cost upwards of $10,000, yet Steinberg says he has no shortage of patients and is currently marketing a new cosmetic option for what he calls 21st century parents-to-be.
25 years ago, I predicted we would be choosing eye color. We're able to do that now. It turns out people want blue eyes. Not only are we able to assist with that, but we can offer them a choice of 30 shades of blue eyes.
These claims are met with great skepticism by many scientists and also raise ethical concerns.
Jeffrey Steinberg claims that he can give you a child with a particular eye color. I don't know what he really means by that, but I think that, again, is an example of how we have to be very careful to draw lines that are clear and can be enforced.
Marcy Darnofsky runs a watchdog group that focuses on the social impact of reproductive and genetic technologies.
What counts as medical? What counts as enhancement? I mean, how could you draw a line?
Today, that question is more relevant than ever.
A medical breakthrough or the first steps down a dangerous road?
In 2017, researchers at Oregon Health and Science University announced a groundbreaking development.
For the first time in the United States, scientists have edited the genes of human embryos.
Using CRISPR technology, they were able to correct a defective gene that causes a potentially fatal heart disease, altering a trait that could be passed on to future generations. They had no intention of using embryos to create babies, but later, in 2019, a Chinese doctor did, and was harshly condemned.
Critics worry CRISPR could be used to create designer babies. Last year, former Director of National Intelligence James Clapper called genome editing a potential weapon of mass destruction, and Congress has banned turning gene-edited embryos into babies.
At least that's what they say.
Dr. Paula Amato is a co-author of the research on editing human embryos.
When you think about the traits that people would like to enhance, things like intelligence or athleticism, we actually don't know the genes that are responsible for those things. And it's likely to be more than one gene. So even if you wanted to do that, at least at this point in time, it would be very difficult, if not impossible, to do.
I think this is a slippery slope that we're on. That doesn't mean that we have to forego everything along the way. It does mean that we have to make sure we have brakes and we have to make sure we have stopping points.
And a 2020 study confirmed the need for caution. Scientists using CRISPR to repair a blindness gene in embryos say it introduced unintended and undesirable chromosomal changes.
All new technologies need to be carefully and properly assessed. I think you can't have the Wild West. On the other hand, I think you can get yourself into a fear situation where you become paralyzed and can't do anything.
When any medical advance is made, there is first of all one success. Somebody had to be first, and then there are others.
There's 6 million of us. Baby's been born through IVF, which is fantastic. And I'm actually quite proud to say that it started with me.
When the polio vaccine was rolled out in 1955, Dr. Jonas Salk, the scientist who developed it with funding from the nonprofit March of Dimes, became an overnight celebrity.
And the entire world heralded the discovery which assured an end to one of mankind's most dread diseases.
Following that, Ed Murrow, who had a television program, interviewed my father, and one of the questions she asked was,
who owns the patent on this vaccine? Well, the people, I would say. There is no patent.
If you look at the original tape of that interview, there's a moment where there's sort of a little bit of pausing. And he thought for a moment, and then you could see the expression on his face just light up. He said,
"Could you patent the sun?"
He just saw this as, this was just a natural evolution of science. The results belonged to the people.
Well, that's the way it was in those days. Scientists had the view that you could either do well or do good, but that it was tough to do both.
AIDS is the most serious epidemic to strike this country since the polio epidemic of the 1950s.
By the time another public health crisis hit America 3 decades later,
the often deadly disease was unknown 5 years ago.
Doctors now say it's a national epidemic.
the pharmaceutical industry had changed dramatically. The costs of research and development had soared, and drug development with expensive clinical trials had become increasingly reliant on investment. Jonas Salk was interested in trying to develop an AIDS vaccine when he was approached by a venture capitalist and asked to collaborate and form a biotech company.
My job was to finance the company. To start on an ambitious effort like the one we did with Dr. Salk, to get the best people working on this, you need to give them the opportunity that this is not just going to be a puff of smoke or a tumbleweed that's just going to blow away.
Salk began to experiment with a vaccine using the AIDS virus itself.
Patents are fundamental to that process. This was not a charity.
And so, Jonas Salk's company ended up filing several patents on a potential AIDS vaccine.
It's not what my father would have chosen. Different world, a different reality.
Today, the pharmaceutical industry has evolved into a $1 trillion business.
It's clear any for-profit company has to answer to both investors, and it has to answer, in our case, to patients as well.
In 2004, Barbara Heiser was diagnosed with a rare form of cancer called chronic myeloid leukemia.
When my oncologist told me what the diagnosis was, my first question to him was, how long am I going to live? And he said, up until about 3 years ago, you would have had 3 to 5 years to live. But he said, now we have this new drug, and he said, you're not going to die from leukemia.
Gleevec has been hailed as a real-life cancer-fighting miracle. But this pharmaceutical marvel has a drawback that is becoming increasingly important in the worsening economy.
Gleevec had a list price of around $26,000 a year when the drug came on the market in 2001. Heiser was getting a deep discount from the manufacturer Novartis, but in 2011, after she changed insurance companies, her costs went up.
So I called to renew my drugs, and the operator said, I'm going to charge your credit card $5,000. And I said, what? At that time, I said to my husband, I said, you know, I feel like I'm being faced with the choice of either keep our house and keep the lifestyle that we have, or stay on Gleevec. We couldn't do both.
Today, the drug can cost over $120,000 a year and has earned Novartis over $40 billion worldwide. Heiser considered the price outrageous, considering that some of the cost of researching and developing Gleevec was paid for with federal funds.
When my money goes to help fund the development of it, and then they're going to turn around and charge me exorbitant prices that I can't afford, then there's something wrong with that.
I think it's important to look at the advancements that we've gotten and ask whether or not those advancements would have occurred without a for-profit industry.
But over the past few decades, innovative drugs have increasingly stemmed from federally funded research. That's because of a little-known law called the Bayh-Dole Act.
This law changed everything.
This 1980 law allowed researchers who made discoveries funded by public institutions like the National Institutes of Health to license their patents to private companies who could develop and market new drugs.
Despite the fact that America was investing a significant amount in research and development dollars, only 5% of patents were ever getting used by the private sector because there was uncertainty around those patents. What the Bayh-Dole Act really did was cause a transformation in terms of the ability of the private sector to utilize that research in the hopes of bringing a new product to market.
That one law said to the big drug companies, you can market drugs, but you don't have to come up with your own original, innovative drugs. That will be done with NIH dollars. And that's exactly what has happened. But do they have a right to be rewarded as though they were the innovators?
And she says the problem with pharmaceutical pricing today extends beyond drugs. In 2016,
a growing firestorm over the soaring cost of potentially life-saving EpiPens.
The price of EpiPens has skyrocketed.
Mylan Pharmaceuticals licenses the patent on EpiPen, a special syringe whose precursor was originally developed with public funds at the Department of Defense that injects epinephrine, or adrenaline, to treat life-threatening allergic reactions.
Over 10 years, the EpiPen has gone from $100 to $600.
The drug itself isn't expensive, it's the delivery method which is.
So Jim Duran led a team at King County Emergency Medical Services that developed a less expensive way of administering the drug.
It would only take about $10 worth of equipment plus the $5 worth of epinephrine to put a package together. We developed a kit, what we call the check and inject kit. So inside the kit we have our little check sheet, 2 safety syringes, and then your bottle of adrenaline.
King County's emergency services saved more than $300,000 a year with this alternative kit. And emergency services around the country also bought kits or learned how to make their own as an alternative to Mylan's EpiPen.
We are going to continue to run a business.
Mylan has since settled a case brought by the government for overcharging for EpiPen. And complaints against high prices have continued.
It's not funny, Mr. Sorelli. People are dying, and they're getting sicker and sicker.
Particularly with some drug companies making record profit margins.
We've seen these prices creep up and up every single year for so many drugs.
And some have pointed to another provision of that 1980 Bayh-Dole law, which allows federal authorities to march in and take over the patent of a publicly funded drug in extenuating circumstances.
The Bayh-Dole Act says the patent has to be made available to the public on reasonable terms. If the patent, drug or whatever else it was, wasn't sold at a reasonable price to the public, the government could march in and lower the price.
There's no history at all that it was intended to address drug prices. I would argue that if march-in rights are used to address drug prices and the government could come in and either give license to someone else or try and control the price, it would undo the positive influence and the positive aspects of the Bayh-Dole Act.
The NIH has declined to use the march-in rule for drug pricing, despite several petitions to apply it to high-priced drugs for such diseases as AIDS and cancer. In 2011, Barbara Heiser decided to solve her drug price problem herself.
I'll be honest with you, the drug that I take has a generic that comes out of India, but that generic is not legal in this country.
Since the Indian version cost a small fraction of the drug price in the US, Heiser started ordering her pills directly.
When I get my shipment, I almost feel like I'm looking over my shoulder to open it up. I feel like a law-abiding citizen has become a drug importer, and that doesn't feel very good.
But she took the risk, since it was the only way she could afford her life-saving medication.
If I were doing it the traditional route, I would pay $1,300 out of pocket. Going through India comes to about $250 for a month's worth.
We have to remember that there are people on the other end of this, and that we not end up depriving people of life-saving medications.
The reasons that I can't get it as a reasonable price from my neighborhood drugstore are all political, and that stuff needs to be fixed.
There was a phrase that my father used, which is more important, the human value of the dollar or the dollar value of the human. Somehow in this complex world, we have to find a mix between the two.